start, let's bow our head and feel the presence of the Lord. In the name of the Father, and of the Son, and of the Holy Spirit. Amen. Dear God, thank you for all the blessings you have bestowed upon us. We are truly grateful for them. Thank you for allowing us today to meet and share our knowledge and time with one another in this webinar. May you extend your divine wisdom to our speaker so that he would be able to impart effectively his God-given knowledge to all of us. May he be blessed as he continues to bring his expertise to people who need them. Bless the participants as well so that they would be able to glean the vital information from this activity. May you bestow your blessings after this seminar so that we may go out and spread what we've learned. In the spirit of your love and generosity, may we realize that this activity should glorify your name. Amen. In the name of the Father, and of the Son, and of the Holy Spirit. Amen.
Mga kababayan, ang pambansang awit ng Pilipinas.
Okay, good morning to everyone. I am very happy to present to you the result of our first seminar on biology, which was conducted last December 13 with our very highly respected speaker, Sir Ruchi Brian S. Lagitnay. So for the objectives of the seminar that was conducted, okay, we have, by the way, we have 198 students who responded on the seminar that we conducted before. So for the different objectives of the activity, were clear and well defined. So it was, as shown on the graph, it is excellent. Okay. And so we have 102 respondents for this, and only 3 responded for the fair. And for the different objectives were attained, and it was also excellent as shown on the graph. For the different activities, so the activity were well organized. So we have 86 responded for excellence, and only 4 for fair. Okay, thank you for that. For the participants were well informed of the activity. So as we can see, 94 responded for excellence, and only 2 responded for fair. So the activity started on time, and again, we are very sorry for this. So we try our best to do better for the upcoming seminars. So only 37 responded for excellence and 18 for fair. So the conduct of the activity was smooth. So 65 responded for excellence, and only 3 responded for fair. So again, thank you so much for the result. The accommodation is good. So 93 responded for excellence and only 5 for fair. The venue where the activity was taken was well organized. So we have 85 respondents for excellence and only 3 for fair. For the speaker, okay, so the speaker shared his expertise appropriately. So we have 122 respondents for excellence. Okay, there is a clear delivery of the activity. So 99 responded for excellence. And the speaker showed good rapport with the participants. So we have 117 respondents for excellence. Okay. Okay, thank you very much, my dear students. And again, hope we enjoy and learned more with our seminar for today.
Hi, good morning, everyone. I'm Tyron John B. Saludares. I'm a faculty of the College of Arts and Sciences here at Vizcaya State University. I'll be your moderator for today. Again for delay. So, welcome to the second lecture of the virtual seminar series in biology, biochemistry, and molecular biology. For those who are new, and for those who were not able to join the first lecture held on Monday, I would like to extend a very warm welcome to all of you. It is our pleasure to have you here again for today's virtual seminar series. Together with the members of College of Arts and Sciences, we proudly present you the second lecture of the virtual seminar series in biology, biochemistry, and molecular biology. So let me introduce the speaker for today's lecture. Our speaker for today graduated Bachelor of Science in Agriculture, major in Soil Science at Visayas State University in 2016. She finished her Master of Science in Soil and Environmental Science from National Chungcheong University, Kaohsiung, Taiwan, last year. She is currently taking her Doctor of Philosophy also from the same university. Let me share you her personal life. Sorry. She started her career in 2016 as a science research assistant for Australian Centre of International Agricultural Research. She then pursued her MS in 2017 and worked as a graduate research assistant and a teaching assistant while taking her master's degree at National Chungcheong University. Her enthusiasm in acquiring new knowledge is what led her in taking master's degree. Just months after she finished her master's degree, because of her desire to share knowledge and become an inspiration to all of us, she worked as an instructor at Cebu Technological University, where she taught principles of soil science, principles of genetics, and biotechnology and society. Her passion to acquire, share knowledge, and reach greater length is what led her in taking her PhD today. While taking her PhD, she is also a part-time lecturer at Eastern Samar State University. She has published several research. Some are under review concerning soil, agriculture, agricultural waste, and heavy metals. She was also invited by different universities for seminar as a research speaker. She is without a doubt a great researcher and lecturer. Today, she will discuss about environmental nanomaterials, current research and methods. Without further ado, let us all give a round of applause to Ma'am Mel Adelle Ocba. Welcome, Ma'am.
Thank you. Thank you, Sir Tyrone, and thank you everyone for having me here to be part of this event. Okay, so Let me start. So again, my presentation is about our laboratory, which is named as Environmental Nanomaterial. So today's presentation, I would like to share our research and the methods that we used to answer the current problems. Okay, so let me show to you. Okay, so we have this logo. So this is our laboratory logo. Yes. Yeah. Oh, okay. Sure. Okay. Should I start again, sir? Or how about this? Okay na po. So I will just continue, sir. Okay. Wait lang po. Ayaw Okay. So this is our logo in our lab. So this logo was created by my lab mate. And my lab mate is currently in Utah State University pursuing her PhD degree. Okay, so for today's presentation, this is the outline. Okay, so I would like to share the three different studies that we have in our lab. And the last part is I'm going to introduce the environmental nanomaterial laboratory members as well as our professor. So first, I would like to share my MS research entitled The Speciation Distribution of Heavy Metals, namely the copper, nickel, zinc, and chromium in the multi-metal contaminated soil remediated by biochar. So just to give you a motivation, I would like to share the motivation why I did this research, kind of research. It is because, okay, heavy metal contamination. This is an extreme deposition of toxic heavy metals in soil due to anthropogenic activities such as mining, smelter, waste disposal from the industry, and intensive agriculture, such as the heavy application of fertilizer. So these activities leads to the heavy metal contamination in soil. So globally, there are more than 10 million sites of soil contaminated with heavy metals. So before, the contamination of heavy metals was in a single metal, but recently due to anthropogenic activities and rapid industrialization, so the contamination in soil of heavy metals in a multi-metal. Okay, that means various kinds of heavy metals in the soil. So let me share the soil contamination in Taiwan. So due to rapid industrialization since year 1980s, so there are variety of heavy metals, soil contamination, and it has become a serious problem. And it has been reported that here in Changhua County, which is close to Taichung, where our school is located, so 184 hectare are mainly contaminated by copper, zinc, nickel, and chromium. Okay, so Changhua County, this county is good for rice production. Okay, so farming of food crops were nearly 850,000 hectare, of which 600,000 hectare are paddy fields. So there's a heavy metal contamination in paddy fields, and it has become a long problem. This is because due to wastewater contaminated with heavy metals discharged produced by the factories and discharged into the irrigation water and used for crop production. Let me share the soil contamination in Philippines, in our country. Actually, there are many heavy metal contamination sites in Philippines, but based on my review of literature, so I just found this, and which are copper, nickel, zinc, and is here. Okay, so in summer, so high concentrations of total nickel were found in an ultramafic soil. Also, high concentrations of total chromium, nickel, and available chromium in red soil near mining site were found. Also, in Manila, so high concentration of copper, zinc, lead, and chromium in the soil were found, as well as in Quezon City, where high concentrations of chromium, nickel, copper, zinc, and lead were found in urban soil. So here also in Zambales and Negros Occidental, so the Zambales and Negros Occidental, the major series there is lowland rice production. Okay, so for us Filipinos, rice production, okay, is very important because, you know, we eat rice for our breakfast, lunch, and dinner, right? Okay, so in Zambales, very high concentrations of chromium and nickel were found, and this concentration exceeded the intervention values in soil. Also, rice plants in Negros Occidental exceeded the toxic levels of copper and molybdenum. And the sources of this high concentration of heavy metals is from the deposition or use of metal-enriched mine tailings and irrigation water. So this table shows the threshold levels in soil. So actually, in our country, we don't have, there is no threshold level set in our country in the BSWM. Okay, so we just used from the paper reviews. For my paper reviews, okay, we just adapted the threshold levels in mainly in Europe from Cabata and Pindias and Cabata Pindias and Pindias 2001. Okay. So about the agricultural waste in Philippines, so about the rice straw and sugarcane. So I want to highlight this one because this is one of the feedstock that we use to synthesize the biochar. Okay, so in Philippines there are it's 300 million hectare, the total land area, of which 10 million hectare are agricultural croplands. So rice and sugarcane are the two most important crops in our country, where rice straw burning is still prevalent at 76% of all rice plants in our country. Actually, rice straw burning in an open field is a big no-no under the Philippine Clean Air Act, but yes, farmers are still burning for the with purpose, of course, okay? But it's actually a big no. So for sugarcane, 64% of the trash at about 3.02 billion a kilogram of sugarcane trash is still burned. So this figure shows the post-harvest burning of sugarcane trash. This is to quickly burn. A farm worker lights on the trashes from one end to the other end of the field. So this photo was taken at Negros Occidental, Philippines. So as we can see, there is a smoke, right? Here. And the burning of the sugarcane. So also for the rice straw. So 90% of the rice straw was generated burnt in the field and only 5% used for other activities. So 10,146 of rice straw are estimated to be born in the field in the Philippines. So this table shows that open field burning of rice straw is not good. Why? Because it produces air pollutants. So these are the pollutants that is produced if you are burning those rice straw or sugarcane or whatever agricultural waste. So this table shows the air pollutant emissions in India, Thailand, and the Philippines. So these are the concentrations of air pollutants emitted from the open field burning of rice straw. So high concentrations of heavy metals in soil is an environmental problem and a long-term risk to human health as they enter into the food chain. Okay, so if our soil is contaminated with heavy metals and if we plant our crops there, so there is a mobility of the heavy metals from the soil. It will go to the crops, and the crops that has been grown in a heavy metal contaminated soil. So if we are going to consume that, it will goes to our body. That's why it's a long-term risk to human health. So about biochar, so in this research, so the biochar is derived from agricultural waste because of that motivation. So why biochar? It is because biochar has a unique properties containing large specific area, porous structure, surface functional groups, and high pH. So multi-metal contaminated soil remediated by biochar has been previously studied. However, little attention has given to the effectiveness of the rice straw, corn waste, rice husk, pineapple leaves, and sugarcane bagasse on the mobility of copper, nickel, zinc, and chromium in various types of soil in the multi-metal condition. For the assessment of metal concentration, so we use the sequential extraction process as proposed by Tesser et al. 1979, which this process provides a precise information on the mobility and bioavailability of heavy metals in soils as well as the sediments. So what is the advantage of this sequential extraction process compared to total concentration of the heavy metals? So here I made a table. So for the speciation, so there is no speciation for the total concentration. So we will not know which heavy metals is mobile or non-mobile. So for the SEP, so this is based on speciation affected by environmental conditions. And for the extraction step, so total concentration is single. It's very easy. It's single. Only one extraction. And for the SEP, this is a multi-step from weakly band to strongly band. Okay, so from the mobile to non-mobile form. And the binding bound, so there is no identity as identity. So as I have mentioned, for the total concentration, we will not know which heavy metals are non-mobile or mobile. And for the sequential extraction process, so we will know the exchangeable fraction, carbonate fraction, iron and manganese oxide fraction, organic matter fraction, as well as the residual fraction. So this figure shows the different fractions. Okay, so from the total metal, so we have 5 different fractions: the exchangeable fraction, carbonates, iron and manganese oxides fraction, organic matter, and residual. And among these fractions, so based from the many research, okay, so exchangeable is the most mobile and bioavailable form. While these 4 fractions, they are dependent on soil properties and environmental factors. So for this research, actually, I had a lot of maybe 5 objectives, but I just summarize it. So these are the main objectives for this study: to investigate the immobilization of copper, nickel, zinc, and chromium by biochar derived from agricultural waste in 5 agricultural soils. So for the biochar preparation, so how did we synthesize this biochar? Okay, so these are the feedstocks that we use. Okay, we have rice straw, corn waste, rice husk, pineapple leaves, and sugarcane bagasse. So these feedstocks were collected nearby Taichung. Okay, and this biochar were pyrolyzed. Okay, so what is pyrolysis under pyrolysis condition? This means which pyrolysis condition means heating in absence of oxygen. Okay, so nitrogen is purged to remove any gas present in the pyrolysis reactor and also to provide inert atmosphere. So for the pyrolysis temperature, we used 500 degrees Celsius temperature, and we have 2 hours per 10 degrees Celsius per minute as the residence time and the heating rate, and 200 ml per minute of nitrogen. So this table shows the biochar properties in terms of yield, ash content, pH, zeta potential, component of biochar samples. This is the unique properties of the the biochar. Okay, so in terms of pH, the rice straw biochar and the pineapple leaves biochar showed a high pH values, and all the biochar samples showed a negatively charged. Okay, and among the biochar samples, the PLB showed a very good element components such as the magnesium, phosphorus, potassium, and calcium. Also here, this table shows the element content and specific surface area of biochar samples. So as we can see, that biochar sample is mainly composed of carbon element. And in terms of the pore size, okay, so PLB showed a greater pore size compared to other biochar samples. And for um, the surface morphology of the biochar, so this was analyzed using the SEM, okay, the scanning electron microscopy. Okay, so as we can see that the surface morphology of the biochar samples are mainly has a porous structure. And for the functional groups of biochar samples, so we analyze it using the FTIR. Okay, we have this one in our lab. FTIR stands for Fourier. That's right, but I forgot this one. Okay, okay. So as we can see that this biochar has a very good functional groups, mainly carbon, hydrogen, and also oxygen. And for the spiking metals in salts, so we do this one using the incubation experiment, and these are the experimental conditions of this incubation experiment. And we use the periodic wetting drying cycles. This is to favor the metal distributions in the soil matrix. And for the biochar treatments in the spike soil, we also do that one using the incubation experiment. And these are the conditions of the incubation experiment. So the temperature is ambient, which is 25 degrees Celsius, and it was in a dark condition. So this table shows the sequential extraction procedure for the speciation. Okay, so we have 5 steps. Actually, it's a little bit time consuming. Okay, so we have here, these are the reagents used, the shaking time, and also the temperature of different fractions. And for the quality assurance, because this is very important, if you're going to, for your study, quality assurance and quality control is important, right? Okay, so all reagents were of analytical grade, and we use this ultra pure water. And for the working preparation of reagents and working standards and also the dilutions, and the working standards for the heavy metals were use the certified standard solution, and blanks and instructions were done in a triplicate, and both QC and both quality control and blank was used to check the accuracy and precision of the AAS instrument. Okay, so to determine the concentrations of heavy metal, we use the AAS instrument, the atomic absorption spectrophotometer. It's a flame by flame. Okay, so also for the quality control and accuracy of the method used in this study, percent recovery was calculated following this formula: the sum of ESP divided by the total metal concentration times 100%. And the speciation distribution was calculated using this formula: the mean fraction divided by the sum of SEP times 100%. Okay, so I will not show you the figures and tables that I have, because it would take maybe 1 hour. Okay, so just I will share to you the mechanisms why the copper, nickel, zinc, and chromium are immobilized by biochars. Okay, so there are 5 mechanisms involved in this immobilization. First is the ion exchange due to improved cation exchange capacity in salts after biochar treatment. In the study, all biochar samples significantly improved the cation exchange capacity in all soil samples. The second mechanism is through precipitation or co-precipitation. This is due to the alkaline and mineral component nature of biochar. So I have in my previous slides, so the biochar, right, has a high pH and it has shows a very good mineral component. Okay, so in this study, soil pH were improved after biochar treatment. Diverse mineral components of biochar were observed in the biochar samples. Mechanism is electrostatic attraction. This is due to the existence of negative surface charge on biochar. All biochar samples showed large amount of negative surface charge. And the fourth mechanism is through physical absorption due to porous structure of biochar. So all biochar samples showed a porous structure using the SEM. So surface complexation, this is due to functional groups present in the biochar. So all biochar samples showed an existing diverse functional groups. So these are the mechanisms involved in the immobilization of the heavy metals by biochar derived from agricultural waste. And these are the results from this study. Okay, so the native soil, which means no heavy metal. So all soil samples rather, copper, nickel, and zinc were in the residual fraction. So residual fraction, these are non-mobile. It will remain in the soil. Okay, and also for chromium, it is mainly bound to iron and manganese oxide, organic matter fraction, and carbonate fractions. However, when the heavy metals are spiked into the soil, this soil, the W, L, R, and F soils, the copper, nickel, and zinc were mainly in the exchangeable fraction. Exchangeable fraction, this is the mobile form of the heavy metal. So in this fraction, no, the heavy metal will go to the plants. However, for the alkaline soil, the fractions are mainly bound to iron and manganese oxide fraction and also exchangeable fractions. And for all samples, generally in chromium, okay, in the chromium, the fraction is mainly bound to iron and manganese oxide fractions. And for the soil properties after biochar treatments, soil pH, soil organic carbon, and cation exchange capacity were improved in all soil samples after biochar treatments. And among biochar treatments and soil samples, highest pH and soil organic carbon content was observed in pineapple leaves biochar and alkaline soil. Highest CEC content was observed in alkaline soil, while in various biochar treatments, highest CEC content was observed in the rice husk biochar, but generally, the CEC in various soil types of soil was observed in pineapple leaves biochar. And for the effects on various soil samples, the exchangeable copper, nickel, and zinc were generally decreased from the control, and the speciation was more bound to carbonate fraction and iron manganese oxide fractions in all soil samples. While in chromium, the speciation was more bound to iron and manganese oxide and organic matter fractions in all soil samples. So let me skip that part. So among the biochar treatments in various types of soil, the PLB generally shows low exchangeable copper, nickel, zinc, and higher OM fractions in chromium. So this could be due to the unique properties of pineapple leaves biochar compared to other biochar samples. PLB shows considerable amount of magnesium, phosphorus, potassium, and calcium, and highest pH value. So therefore, it is concluded that PLB shows a great potential in the remediation of heavy metal polluted soil. So after biochar, in comparison of various metal, the effects on various metal, so after biochar treatments, the cation metals in the exchangeable fraction in various types of soil were in the following order: copper, zinc, and the nickel. So, however, For the nickel, some biochar treatments and soil samples, the exchangeable fractions were increased from the control. So thus, considerable attention should be paid in the remediation of nickel in a multi-metal contaminated soil using biochar. So that's the end of my research. Okay, so let me proceed to the next study from my lab mate. Okay, so that her research is entitled Agri-Biodegradable Mulch Films Driven from Agricultural Waste on Sustainable Agricultural System. Okay, so this presentation, actually she presented, my lab mate presented this presentation last November 18, 2021, in the 2021 Thailand-Taiwan Bilateral Symposium on Science, Agriculture, and Technology. Okay, so this research is in collaboration in one of the universities in Taiwan and also in Thailand. So just to give you the highlight of this research, okay, so this study was successfully synthesized the biodegradable mulch for the application of soil amendment and plant cultivation. So the soil organic carbon, the nitrogen and phosphorus were improved after being treated by the biodegradable mulch. So the mulch could enhance the application of fertilizer to the soil as evidenced by the plant growth, and the improvement of soil quality and plant growth trials confirmed that has the ability to release fertilizer of mulch after being degraded. So this is the motivation of this research. So biochar can improve the physicochemical properties of soil. So biochar able to release nutrients and the immobilization and release of nutrients in soil from the biochar. Biochar can improve the microbial properties in soil. Okay, and the other one for the shrimp, for the chitosan. Okay, so annually there are more than 6 million tons of shrimp waste are produced in China. A large part of the shrimp waste is often dumped in a landfill or the sea, which leads to serious environmental pollution. However, this shrimp waste contains chitin, protein, and calcium carbonate. So that's why we will use this shrimp waste as our material in order to be used in the agricultural system. So for the poultry, so poultry industry is expanding rapidly and producing million tons of feeder waste annually. So there are two main feeder disposal methods. So these are burning and burying. So both of these disposal methods have a negative impact on the environment. So we will use this. We use this chicken feeder not to make us a new material. Okay. So for the plastic molds residue pollution, so we all know that this plastic molds, right? This is an agricultural problem. One of the agricultural problems. This is because it may cause soil degradation and may post plant growth impact. Okay. And for the novelty of this research, okay, so the biodegradable polymers, so the breakdown of such mulches in the natural soil environment is low. And the novel biodegradable mulches that we have synthesized, no, coated with biochar, shrimp waste, shrimp shell, and chicken feeder have multi-functions favorable for the plant growth. So the fabricated biodegradable mulch was derived from different agricultural waste designed to be degraded in the agricultural soil. So these are the objectives of this research: to synthesize the biodegradable mulch from the agricultural waste, to evaluate the soil quality after mulch degradation, and the third one is to study the fertilizer releasing performance of biodegradable mulch on the plant cultivation. Okay, so these are the agricultural waste used. So we use the tea, pineapple leaves, the coconut fiber, cassava stem, and the palm branch. And for the chicken feeder also, all right, as I have mentioned, the shrimp shell. So these are the materials, the biochar, the hydrolyzed keratin from the chicken feeder, and the chitin from the shrimp shell. So chitin, it contains nitrogen, phosphorus, and calcium source. And for the chicken feeder, it it can be a nitrogen source, and for the biochar as a carbon source. So these are the materials and methods. Okay, so for the biochar or biodegradable composite properties, so we use these instruments, the FTIR, TGA, the tensile or flexural mechanical, and the SEM for the morphology of the samples. For the biochar synthesis, so we synthesize this biochar, and these are the characterizations, the ultimate analysis, the physicochemical analysis, and the surface analysis. And here for the application of the agri-biodegradable mulch. So we have here the control group. Control group is without mulch, and PLA, which is the bioplastic, and the agri-biomulch film. Okay, so we use the bioplastic plus the biochar, the shrimp shell, and the chicken feeder. So here, to determine the K functionalities of mulch, so we characterize the prepared agrobiomulch film, and also we analyze the biodegradation using the soil physical, chemical, and biological properties. So this table shows the characterizations of the mulch. So as we can see that the specific surface area, okay, ranges from 1.64 to 1.05 to 3.67 milligram. Yeah, that is for the specific surface area. And in terms of the exchangeable bases, so the prepared biomulch contains very good exchangeable bases, as also it contains a high pH values. This figure shows the surface morphology of the prepared mulches, biomulches, biodegradable mulches. So as we can see that there is a porous structure of the prepared biodegradable mulches. And for the element components using the EDS analysis, as we can see that all the biodegradable mulches is composed of mainly composed of carbon element. Also, for the functional groups of the biochar and mulches, as we can see that the original biochar is mainly composed of carbon element, right? And also for the biodegradable mulch. So for the soil properties after biochar mulches treatment, in terms of exchangeable cations, it is very clear that the sodium, magnesium, calcium were improved after the application of the biodegradable mulch in comparison with the control. Also, for the organic matter and organic carbon, the soil pH and electrical conductivity, the soil phosphorus and nitrogen, as we can see that there is an increase of these soil chemical properties in comparison with the control. Also, for the soil enzyme, so this figure shows the soil enzyme activity after 28 days of mulch treatment in alkaline soil under the greenhouse environment. You can see that the enzyme activity is increased, has a huge number compared to the control. And for the soil moisture under the biodegradable mulch during the growth of the lettuce, as we can see that with the application of the biodegradable mulch, the moisture were improved compared to the control. So that means the biodegradable mulch may preserve the soil moisture. Also for the soil temperature. So the soil temperature were improved after the biodegradable mulch were applied in the soil. So for the soil carbon stock, okay, so as we can see that the carbon content were in the soil were improved after biodegradable mulch is applied. So in here, this figure shows the mulch deterioration and plant cultivation at 7 days, 14 days, 21 days, and 28 days. So as we can see that in comparison with the control and only the bioplastic film, okay, as we can see that it's very clear now that the plants are growing well compared to the control. Also here, it is more obvious at 28 days that the plants are growing well with the application of the biodegradable mulch. Also this one. So for the plant growth measurement, the root shoot, the fresh weight, and dried weight were determined. And as we can see that in terms of the fresh weight, so the application of the biodegradable mulch greatly improved the fresh weight of the plant as well as the dry weight. And here is the mulch's residual before and after harvesting. As we can see that after harvesting, the biodegradable mulch um, it's already starting to degrade. And for the surface morphology after biodegradable mulch has been applied, okay, so as we can see that the biodegradable mulch has been degraded. So this is before and after 35 days. It is very clear, right, that the biodegradable mulch has been degraded in the soil. Also here, the EDS analysis before the pineapple leaves biochar molds were applied after 4 months. So as we can see that before the element, these are the elements, and after the treatment, okay, so these are the elements left in the biodegradable mulch. That means these elements are consumed by the plants or by the crop. Also for the weed growth, so it is very clear, right, that in the control there is a weed here growing, okay, and there are no weeds in the with the mulches treatment. Yeah. So to conclude this study, okay, so the agricultural waste were successfully prepared, and the characterizations of the biodegradable mulch has been identified. Okay, so the biodegradable mulch, so this is a carbon sink in the soil. Okay, so that's the end of this research. So let me proceed the next study in our lab, which is the disinfection, about the disinfection kinetics and mechanism of A. niger. So A. niger is a fungus, and K. pneumoniae, using K. pneumoniae is a bacteria. So the disinfection is treated with chitosan and calcined oyster shell. So this is also a research study of my labmate. Okay, Tantinuan. Okay, she also joined a conference recently. Okay, so this one is in Chinese. Okay, so this is the motivation for this research. So the fishery problem. So there are 600,000 tons produced for the shrimp waste and 6.3 million tons for the oyster shell globally. So in Vietnam, so because my lab mate is from Vietnam, so there are 530,000 tons per year of the fishery waste, and in Thailand, 620,000 tons per year, and in Taiwan, 480,000 tons per year. So we have this infection issue, right? And there are 3.5 million infections in the hospital due to bacteria or maybe, yeah, the fungus. So the solution is that the fishery waste from an derived novel antimicrobial materials, okay, derived from chitosan and calcined oyster shell. So in this presentation, I will discuss about the mechanism about this antimicrobial properties of these materials. So the novelty that there is a disinfection performance of K. pneumoniae and A. niger via calcined oyster shell and chitosan was first studied. So K radicals for the COS and CTS disinfection mechanism also identified. The biophysical properties and 3D tomography of the disinfected K. pneumoniae and A. niger were also identified. And these are the objectives of this research: to synthesize and characterize the environmental friendly calcined oyster shell and chitosan from fishery waste, to investigate the disinfection performance of the different microorganisms, the gram-negative and the fungi, to study the kinetics of disinfection capacity of the COS and CTS, and to identify the K radicals in the disinfection mechanism. And the last one is to explore the biophysical properties of the inactivated cells during this disinfection process. And these are the materials and methods used for this research. So in this study, applying popular methods such as the SEM, the scanning electron microscopy, the TEM, and AFM is used as an excellent tool, because the AFM methods, okay, for the AFM methods, these are observed in a natural strategy without scanning or conductive coating in vacuum. And for the 3D tomography or the TEM, the technique this technique is used to identify the cellular structure deformation at the beginning of the lag phase. Okay, in the next slides, we will see the result, how this 3D tomography uh, will give us the good result, as well as the AFM methods. Okay, so this is the soft X-ray tomography in the NSRRC here in Taiwan. So NSRRC is located in Hsinchu in the northern part of Taiwan. So the soft X-rays within a region of the spectrum known as the water window can penetrate cells that are up to 15 micrometer thick, indicating it can require a 3D image from nearly native thick cells without the end of staining and sectioning of the specimen. So the sample target here is the bacteria, okay, the cell and also the tissue for this instrument. So let's proceed to the results. So these are the characteristics of the calcined oyster shell and the chitosan. So these slides give us the information of the COS before and after heating treatment. So in comparison, from left to right, okay, the COS has been successfully prepared from the oyster shell, that the ground oyster shell particles have a layered structure with a rough surface. And before heating, the COS has irregular and almost centered and shaped particles. Okay, so the COS and CTS have been preferred from the fishery waste. So here, the first experiment is the different dosage effect. Okay, so the gram-negative bacteria, the KP, and the fungi, A. niger, are the microorganisms modeled in this research. So each microorganisms use 5 different dosage of the COS, and it is easily found that here for this result that KP is more sensitive compared with A. niger. And when the dosage of the COS is increased, the reaction time will also increase. This will lead to the disinfection efficiency also increase. And at the highest, sorry, and at the highest dosage, the COS can kill the KP within 60 minutes, while A. niger is 120 minutes. And for the microorganism concentration effect, okay, so this experiment is different dosage. Okay, so for the antimicrobial activity, though the higher concentrations of microbes requires a longer reaction time of the COS and CTS attacks to sterilize. So at all concentrations of the experimental microorganisms, both the fungi and the bacteria, the CTS and COS showed antibacterial activity up to 99.999%. Yeah. And for the disinfection kinetic model, so we use this model to describe the disinfection performance of the prepared materials, the CTS and the COS. And these are the result. And we use the modified home kinetic model to fit the kinetic the survival of the bacteria. So for this disinfection mechanisms of the, so I will discuss separately the mechanism of the COS and the CTS. So here we analyze the change in morphology and the ultrastructure to see the difference. Okay, so later on we will see the morphology, the reactive species, the cell permeability, the biophysical properties, and the 3D cellular structure. So in here, using the scanning electron microscopy, it shows that COS can break the outer membrane of the A. niger that also can be found by the TEM at 2 hours. However, after 6-hour treatment, the A. niger has lost the formation of the organelles, leading to cell death from inside. So this one, the K. pneumoniae and the A. niger has been compared using the TEM and the SEM. So there is a difference, right, between the two instruments. And for the survival of bacteria, so I have already mentioned this one. So the COS can penetrate the cell membranes and the intracellular structure has been damaged, leading to the disinfection of the A. niger and K. pneumoniae. So this is because of the oxygen, active oxygen. And for the cell permeability, so in here, the double staining, the DAPI and PI were used to define the live cell and dead cell. So of the K. pneumoniae, so as expected, the number of red points increased after 6-hour treatment in here. Moreover, the survival rate of bacteria count by staining ratio, the green one, and NA plate culture, the red one, has been also compared, and the results shows that there is a change of the cell permeability, which is the main reason why the cell is okay. So that's it. here. So the cell permeability has been damaged and impact the cell structure. And for the biophysical properties, so we use this one, the 3D images from the AFM, which shows that the KP cell structure is collapsed, while the outer part of the A Niger is totally broken, which is similar with TEM and SEM results. So this is the height, the roughness, the DMT, and the adhesion of the two microorganisms. So this is the very interesting part. Okay, so the 3D tomography and the 2D SEM image were compared. Okay, so as we can see, okay, the A Niger after 6-hour treatment, okay, has been damaged. Okay, so let me show this. So this is the SEM image. So still showed a spherical shape after 6-hour reaction. And for the 3D tomography, so this is the control. This is the A Niger without treatment, right? So the cell is not damaged. It's still active, okay, of the A Niger. And when it is treated with CUS at 3 gram per liter at 6 hours, you can see that the cell has been damaged. Okay, so this is how the 3D tomography works. Okay, so 3D structure show the cell colonies of the A Niger which is damaged under the CUS treatment. Okay, almost done. So these are the summary of the disinfection performance of the different disinfectant and the disinfectant that we have synthesized in our laboratory. So in terms of the disinfection performance, so it shows 99.999% for the A Niger at 2 hours and 99.999% for the K pneumoniae in 1 hour. And in comparison with the previous studies, so they don't show the disinfection mechanism, but in this research, the disinfection mechanism has been provided. That's the novelty of this research. Okay, and for the CTS, sorry, it's just the COS. For the chitosan or the CTS, so we analyze the change in morphology and ultrastructure to see the difference. So in here, this figure shows the morphology and ultrastructure of the microorganisms using the TEM and SEM. So in the absence of chitosan derivative, the transmission electron micrograph of the K pneumoniae, okay, so this has been damaged right after 6-hour treatment. And this figure shows the negatively charge of the chitosan to disinfect the A Niger and the K pneumoniae. So chitosan interacts with the membrane of the microorganisms to alter the cell permeability. And for the biophysical properties, using this 3D image from AFM, it shows that the KP cell structure is collapsed, while the A Niger is totally broken, which is similar with the TM and SEM results. So this is using the chitosan treatment. The destruction of cell walls and membranes appeared to play an important role on enhancing the disinfection efficiency. Okay, that's why we have to use this 3D image from the AFM. And for the 3D reconstruction of the A Niger using this CTS, okay, so as we can see that after 6-hour treatment, the 3D structure show the cell damage when we rotate the cell in different angle. So the good about this 3D reconstruction is that we can see the the cell structure, the damaged cell structure after the treatments. And for the using the SEM only, we cannot see that one. So let me show you this one. Okay, so this is the structure of the A niger. And after the treatment, okay, so as we can see that some cells are already damaged after the treatment. And this infection summary for this chitosan, okay, so the 99.999%, there is this is this infection rate for the A niger within 2.5 hour, and for the K pneumoniae, so also 99.999% at 0.5 hours. So in terms of the disinfection, the K pneumoniae is more faster compared to the A niger. Okay, so to conclude this this presentation, okay, the synthesize and characterization of the COS and the CTS. So the environmental friendly COS and CTS have been successfully synthesized from the waste shrimp and oyster shell. The chemical composition, the chemical binding, surface functional groups, and the morphology of the COS and CTS were identified via ESR, XRD, X-ray diffraction, FTIR, and the SEM. So for the disinfection performance and kinetics, so both the C2S, COS, and CTS are highly effective in disinfecting these microorganisms, and 99.99% rate were achieved for the disinfection of the K pneumoniae within 0.5 hour, and for the A niger is the disinfection time is within 2 hours. So different compositions of the cellular membrane of the gram-negative bacteria, the K pneumoniae and A niger, leads to the disparate disinfection efficiency. Disinfection kinetics is appropriately described using the modified home models, showing a good prediction of the disinfection performance of the COS and the CTS. And for the disinfection mechanism, the singlet oxygen is the major radicals that plays an essential role in the disinfection mechanisms of the COS. And for the chitosan, the cationic amino groups on the positively charged chitosan interact with the negative charged cell membrane, which increase the membrane permeability and membrane lysis. For the biophysical properties and the 3D tomography, the biophysical properties such as the cell height and the DMT modulus of the inactivated K pneumoniae and A niger were decreased with disinfection time. The surface roughness and adhesion increased with an increasing reaction time. So the TXM and the SXT successfully obtained the 3D tomograph of A niger at the nanoscale, which shows the potential of early diagnosis. The disinfection kinetic and mechanisms of the COS and CTS are obtained and both clarified. I hope it's clarified. Okay, both CTS and COS have the high potential to alleviate the aquaculture waste pollution problem globally, and this can be applied as a highly efficient disinfectant. So that's all for my presentation. And for this research, the last one that I discussed. So this receives huge awards and publication from the international and local conferences, and these are the ongoing publication for that research. And the last part, I would like to introduce and acknowledge my professor and also the lab members in the E&M. So this is my professor, Professor Yao Tong Lin. He is a distinguished professor in Institute. This is our collaborating professor from the Issue University in Kaohsiung, and we have Dr. Yan from Vietnam. He is our postdoc in the lab, and we have Brooke, Mila, PhD. These students, Brooke and Mila, actually they are PhD dual degree. So Brooke is currently in the University of Delaware in USA, and Mila is currently in Utah State University, and also Cassini and Nook from Thailand, and Nook and Bubu from Vietnam, and me. And we also have Jack from Taiwan. He is pursuing his MS degree, and he continue my MS research. And we have also Janice, and we have a new lab member from Philippines, Jerickson. He finished his bachelor degree in CLSU, and also we have Jane. And that's all for today's presentation, and I hope that you learned something from me. Okay, if you have any questions, feel free to ask. Thank you.